Hello, I'm Kwang and I'm working for Ewha Women's University Seoul Hospital. And this is great honor to have an opportunity for interview from ICUrology. And today I will introduce my paper, Urinary Microbiome Profile in Men with Genitourinary Malignancies. Traditionally, urine was believed as a sterile condition, and we know that urinary tract was not included in human microbiome project, which started in 2007. However, if we use advanced culture technique, we know that bacteria can be isolated from urine, which had been reported as no growth in standard culture technique. Also, if we use NGS technique, we can analyze DNA fragments and we were able to identify the presence of bacteria in the urine. Several studies have investigated the urinary microbiome in urologic cancers and have revealed a different urinary microbiome composition in urologic cancers, but the results were not consistent and none of the prior studies have analyzed multiple urologic cancers at the same time. So in this study, we aimed to characterize the urinary microbiome in three major urologic cancers, including prostate cancer, bladder cancer, renal cell cancer, compared to that of patients without malignant disease. This study was conducted under the approval of IRB, and for this study, first void urine samples were collected on the day of surgery. DNA was extracted from supernatant of urine, and 16S rRNA sequencing was performed, and the bioinformatic analysis was performed as described in this flowchart. We initially started with 104 male patients, and after quality control, 19 samples which had sequencing lead less than 5,000 were excluded. So 85 samples were included for the final analysis. When you look at the alpha diversity, prostate cancer and renal cell cancer had a significantly lower index than bladder cancer, indicating that urinary microbiome of prostate cancer and renal cell cancer may be dominated by a few abundant bacteria. Also, Palmanova analysis based on Bray Curtis showed significantly difference in urinary microbiome composition among the four groups. In 1A ANOVA, 6 genera including Cutibacterium, Peptonopilus, Spingomonas, Staphylococcus, Micrococcus, and Moracella were significantly different in abundance among the four groups. In species level, we compared each urologic malignancies with non-cancer group by step analysis. When we compared bladder cancer and non-cancer group, 7 species were more abundant in the bladder cancer group, and the difference was most pronounced in Micrococcus species. We also identified 18 species in prostate cancer and 8 species in renal cell cancer that showed different abundance from non-cancer group. One of the species of interest is Cutibacterium acne, and Cutibacterium acne is associated with infectious skin disease such as acne vulgaris, and the association between Cutibacterium acne and prostate cancer has long been discussed, and we know that recent molecular study found that Cutibacterium was the most abundant genus in the radical prostatectomy specimen. And one epidemiologic study from Sweden also found that a diagnosis of acne in late adolescence significantly increased the risk of prostate cancer. In our study, a significantly higher abundance of Cutibacterium acne was observed not only in prostate cancer but also in the renal cell cancer group. And interestingly, 4 species, including Cutibacterium acne, were significantly abundant both in prostate cancer and renal cell cancer. So, in conclusion, major urologic malignancies has a different urinary microbiome composition, and we identified bacteria which are significantly associated with each type of urologic malignancies. Specifically, several bacterial species were associated with both prostate cancer and renal cell cancer, suggesting that both cancer may share a similar pathogenesis related to urinary microbiome. Finally, I think one of the major limitations in analyzing the urinary microbiome is that the absolute quantity of urinary DNA is very small, and moreover, of this small volume of urinary DNA, most are from human host and very small proportion is from bacteria. And of this small volume of bacterial DNA, most bacteria is less than 1%. So I'd like to say this is completely different from analyzing fecal microbiome, which had lots of bacterial DNA. So this is very sensitive experiment, and the results can be changed by a very small difference in experimental conditions such as sample preparation, PCR amplification, or sequence condition, etc. So I think that's why the results were not consistent in the previous studies. So actually, this study began about 4 years ago, and during that time, many methods were changed, and I have also performed sequencing twice to correct the error and analyze the results multiple times using the different bioinformatic pipeline. And in the last year, International Consortium for Urinary Microbiome Stone Disease made a kind of standardized protocol for urinary microbiome analysis. I also agree that future studies should follow a standardized protocol for urinary microbiome as far as possible, and they can lead to more consistent results that can be replicated by the other researchers. Thank you very much.